Okay, I'm recording. A couple of announcements. I talked to Dr. Reavers about the idea of what if some students want to get into the lab outside of our normal lab hours to do their research project. And he sent me back an email confirming that there would be certain times that there would be somebody present. So the only way we can do it right now is if somebody is present in the lab to be with the students. So uh, Alex will be there on Tuesdays from 2 to 4, JP from 4 to 6 on Fridays, and then Rachel on Wednesdays from 2 to 4. So I'll post that into the announcement section in EduCat as well. So those would be times that you can go in once you get cooking along in your research project if you need some extra time and want to check things. Of course, the limitations are that you have to have only 12 students in that classroom at a time or in that lab. So 12 people, 12 students and the instructor or the monitor person. So if you, I think it's kind of first come, first serve. If you show up and there's more than 12, I asked Dr. Reavers if students could go into the next door room, we call it the core lab, right next door. And he said, yeah, if there's two people in there, apparently they want to limit it to 2. And sometimes that core lab is used for prep work. It's not really an official class lab, but that would be an option. And then maybe even the room beyond that, the biochem teaching lab, that would be another option, I would think, if there's no one in there. You could take your plates and go in there to, inspect them or whatever you need to do. You could probably wheel a microscope over there too. So there are options, but they need to kind of keep an eye on everybody, especially with the COVID situation. So that's the situation with how to get into the lab if you want to after our normal lab period. And then the outlines due today. So when we go into lab today, it's group 1, and I can talk with you and help you get going, answer any questions. But remember, the outlines due for everybody today, and there's a Dropbox in EduCat. And then the other announcement, Wednesday, we normally have lecture again. I'm going to cancel Wednesday's in-person stuff. I have to attend a funeral here in town. Family member passed away, so I will be going to that on Wednesday. And I think what I'm going to do is tomorrow I'll just do a Zoom recording of my lecture of what we would have done that day, and I'll post it, and then you can watch it at your leisure sometime on Wednesday. Okay. So Wednesday, no lecture. We don't have lab that day, so it's just the lecture that I'm going to be canceling and posting a lecture. Okay. So I think we left off around here, looking at the idea of plants and microbes. And there's so much stuff to say that I could spend the whole semester talking about this, but more and more now they're recognizing how important the microbes are for plants. And I think I alluded to this last time we talked, that they think it's mainly because of the human microbiome project that really kind of pushed everyone to start thinking about these interactions between microbes and their hosts. And I like this quote that this article had that I put up here with root systems, which absorb nutrients and are directly exposed to soil microbes, are functionally analogous to the animal gut. And some people take issue with that. I think it's a pretty nice way to think about it, though. Some people are a little too picky about it and say, well, the roots are outside into the soil. It's not an enclosed GI tract. And yes, that's true. But it really is the main interface that plants have with their microbe partners. Of course, they don't have an internal gastrointestinal tract. So this really is a dynamic area. The root systems are heavily colonized by microbes that hang out attached to the roots. We'll talk about some special cases, but you have to realize there's just, normal bacteria and fungi that hang out on the roots, as well as the mycorrhizal fungi we'll talk about, and the nitrogen fixers. And so it's just it really is analogous to the animal gut in the sense of that's the part of the plant body where you see huge populations of microbes that interact with the host and do a lot of beneficial things. Certainly, pathogenic microbes can gain a foothold too, but there's a lot of beneficial microbes that can influence the plant in a very positive way. So this is I scanned the cover of a book. They write whole books about this concept here with the mycorrhizal fungi. Conferences devoted to just mycorrhizal fungi and their partners, the host partners. So there is a very active area of research with regard to this because we recognize there's so many plants, vast majority of plants have mycorrhizal relationships, and mycorrhizal fungi have been super important all the way from the get-go when plants started making it into the terrestrial landscape of Earth millions and millions of years ago. So you're looking at a cute little conifer seedling that's growing quite young, and you can see its tannish roots, and then you can see the mycorrhizal fungi coming off of that, and you can see they're linking. There's a couple other seedlings here on either side of the main one, and you can see that they're linking together. That's another really, really important concept that they're really chasing down and trying to figure out in forested ecosystems, looking at how the mycorrhizal fungi, they call it the web essentially, and that it's connecting all of the trees and plants in that area, and lots of communication and exchange of chemicals. Here on the right, you're seeing another conifer seedling. And again, the vast majority of what you're looking at is hyphae of the fungi. And so these are the fungal hyphae strands that are growing off the root and into the soil system. Here's another excellent picture showing the roots as the larger, darker on here. And then you can see the mycorrhizal fungi, the hyphae extending off. And so, I don't know why I put FV, that was a typo, visualization of the intact. So here you see the idea of glomus is a certain type of endomycorrhizal fungi that we'll talk about in a little bit. And that one is super popular because it has a broad host range and can really help plants grow. A lot of people put that into their garden as well if you're gardening. So glomus species in particular have been heavily looked at. Okay, so what are we talking about here? Mycorrhizal fungi. Mycorrhizae, that term literally means fungus root. If you look it up, myco is the fungus, rhizae root. And so it is, when you say mycorrhizae, you're talking about the roots that have already been established with the fungus. They already are in a symbiotic relationship. And then if you talk about the fungi just on their own, you call them mycorrhizal, Z-A-L, mycorrhizal fungi. And so it's the vast majority of plants have this. It must be a win-win situation for this dynamic to be coexisting for all these millions of years. And so the fungus does get carbs, certainly from the plant, and they've done some nice tracing where they look at the carbon that the plant takes in and does photosynthesis, makes sugars, and then where does all that sugar end up? And they can show that they did some studies in forested ecosystems where 30 to 40% of the fixed carbon that the plant makes goes to the fungus. So it's a significant investment on the plant's part in that respect, that they are feeding these mycorrhizal fungi with sugars. And so you would think if they're going to do that, they're going to get something back in return, and they certainly do. The plant gets increased access to water. If you just think about the mycorrhizal hyphae, they're so much smaller than even the smaller, finer roots on a plant. And so the hyphae are nice and small in diameter. They can creep and grow into cracks and crevices of the soil system that the roots just physically cannot access themselves. So of course they can get access to some water that the plant just can't get to. And then I put this in ranked order, PNK, that's a ranked order. Phosphorus in particular, the fungus is really good at getting phosphorus solubilized and getting it taken up from the soil system and getting it to the plant, and also nitrogen and potassium. So they've done this through controlled studies in the lab. They've looked at it in, actual ecosystems, and they can show that this is all happening on behalf of the fungus. And so, in fact, you have to be a little careful when you're working with mycorrhizal fungi. If any of you are going to do an experiment with mycorrhizal fungi, and I've had this experience with my grad students and then students in plant phys that are working with mycorrhizal fungi, you can't be too nice to your plants if you want them to form that relationship because we were one of my first grad students. She was working with a certain type of ectomycorrhizal fungi, and she couldn't get the partnership to form. And they grow really slow. So 6 months later, when you dig up the roots, and they're like, ah, darn, they're not even forming a relationship. So that was a downer because she, 6 months of her life gone. And then she realized after we spoke about how she set up her watering and fertilizing regime that she was being too nice to the plant. She was giving the plant too much phosphorus. And so the plant just said, I have enough phosphorus. I don't need this partnership. And so the plant never engaged in forming a relationship with the fungus. So then the next round, we stressed the plant, gave far less phosphorus, just a little bit, and stressed it a little bit with water. And then boom, we got lovely exomycorrhizal fungi. So you have to be a little careful when you're designing experiments not to overdo it with the plant. They can actually, because a lot of phosphate is complexed with other chemicals and then it precipitates out, and they actually can have the enzymes necessary to break those compounds apart and then release the phosphate, and then it can get into solution. So there's a lot of different forms of how phosphate can complex, but fungi in particular, mycorrhizal fungi, are really quite good at solubilizing all that phosphate and getting it released into the water. And that's what the plant needs. The plant needs to have things dissolved in water before it can take it up and use it. And there's well over 1,000, if not many thousand papers published showing if you have plants in a lab situation where you deliberately deny that plant mycorrhizal fungi and then you set up another same plant with mycorrhizal fungi, huge difference in their growth rate, the plant's growth rate, its biomass at the end of its growth if you're harvesting it for food production, for example. So it definitely gives the plant a boost right from the start, right from the seed stage on through to maturity. Having that fungus there can really stimulate growth and make a big difference in its overall biomass and growth rate. And then it can protect. There are mycorrhizal fungi that are good at defending the plant against plant pathogens. And so we see that it can be done chemically, where the fungus is releasing some kind of antibiotic into the soil system that can prevent the pathogen to do any significant damage. Physical, though, some mycorrhizal fungi form a nice physical barrier, so we'll look into that. And so it can be both chemical and physical that it's doing this job of protecting. And then there's lots of studies looking at what else can the mycorrhizal fungi do. And this is just a tiny list of what they've been investigating. The list actually goes on, but they're looking at all kinds of ideas of if you can find the right partnership with the fungus and the plant, you can see some nice growth in really droughty soils that are marginal in terms of they've always been like, yeah, that land could be used, but it's so droughty, most plants can't handle it. And now they're finding that if you find the right partnership between the fungus and the plant, you can use some of these marginal, less than desirable lands. Heavy metal, they've looked seriously at this. So the fungus has the ability to take up heavy metals that are toxic to plants, and they're toxic to us too. But they can take up the heavy metals and then sequester them in their fungal hyphae. So the plant doesn't get access to it. It's contained within the hyphae, and then it stays there. A group of undergrads that I worked with just, I guess it's 2 years ago now, they did a nice study with the zinc heavy metal and looking at the presence or absence of mycorrhizal fungi in their host plant was lettuce. And with the idea of, if we grow lettuce in marginal land that might have heavy metals, can we eat the lettuce or are we going to get exposed to heavy metals? And so they were looking at what happens when you mix in the mycorrhizal fungi into this system. And they nicely showed that the presence of the mycorrhizal fungi decreased the zinc in the soil. So it was cleaning up the zinc from the soil, but it didn't end up in the tissue to a huge extent. It did in some concentrations of the mycorrhizal fungi. But basically, what they were showing is the fungus was good at remediating the soil and sequestering some of that zinc into its hyphae. And so that was an interesting. They got that published. It was a really nice study. So this one, looking at again, you see images like this in the published literature over and over and over, looking at the start of plants from the seed stage, and so these are young seedlings without on the left, without any fungi, and with, and that's very, very typical. That kind of dramatic difference in growth right from the start. So there's different kinds of mycorrhizal fungi, and it just depends on what kind of host plant you're looking at and what they have evolved to partner with. And so it's kind of hard to read the black, isn't it? I should change that to white. But so here on this table, you're looking at, on the left column, different groups of mycorrhizal fungi. And so the vesicular or buscular mycorrhizae are called, you shorten that down to VAM, B-A-M, VAM. And the other word for them is endomycorrhizae. That means the same thing. And then there's the ecto. So some people prefer the endomycorrhizae and the ectomycorrhizae because it's kind of a nice comparison. So we'll talk about those, and then we'll also mention a couple of these other ones. So especially the orchidaceous mycorrhizae, they are real interesting as well. And so I've asterisked up here in the middle column with the VAM fungi. It says fungi produce hyphae, arbuscles, and vesicles in plant root. So that's kind of vague. I want to make sure that you understand that they're not, the fungal hyphae aren't swimming around in the cytoplasm just free. They're always surrounded by a plant-derived membrane. So the plant is always keeping the fungus compartmentalized so that they're not just swimming free, so to speak, in the cytoplasm. And that is what they refer to as a controlled infection. They say it's a controlled infection, that the plant is really controlling how far the fungus can take it in terms of the intimacy. And respect my boundaries. I'm not letting you swim around in the cytoplasm. There will always be a plant-derived membrane around the fungus, and so that prevents a triggering of the plant defense response. Because if a pathogen or if a fungus gets into the cytoplasm, that typically stresses the plant, and they start to defend themselves against it. They consider it a pathogen. And then on the right, you're seeing different categories of fungi that form these relationships. So if you've ever learned about fungi in BI 112 maybe, or if you had the luck to take the mycology course with Dr. Robertus, I'm sure you've heard these terms before with the different groups of fungi. So when you look at the ectomycorrhizae, if you're out hiking around, hopefully you got out this weekend in between the snow and the rain, and we're out hiking in the woods and saw some beautiful color changes. Maybe you still saw some fungi, some fruiting bodies, some mushrooms around the plants. And so typically when you see those fruiting bodies around here, it's a good guess that they're ectomycorrhizal fungi that are making those fruiting bodies, and that what you're seeing is just a tiny, tiny example of the fungus. The fungus biomass is huge below ground. So every time you're out walking in the forest, you're walking on literally kilometers and kilometers of fungal hyphae beneath you. And you just, unfortunately, we cannot witness that with just hiking around. And so most of the trees around here, most of the shrubs around here definitely have ectomycorrhizal fungi associated with them. And they can, depending on the species, also have endomycorrhizal fungi associating with them, but for sure a range of ecto. So endomycorrhizae or VAM means the same exact thing. It just depends who's talking. These fungi are quite a bit different in some respects, so we'll work through this. But they tend to not show up too much with trees and shrubs. There's some exceptions, but their main host are the so-called non-woody plants, the ones that don't produce, true secondary wood. So, if you think about maple trees and oak trees, they don't really hang out with those types of plants. But there's some exceptions. But and then the angiosperms in general, the flowering plants, there's a lot of different endomycorrhizae that hang out with the flowering plants, certain species, mostly non-woody. And then the orchidaceous mycorrhizae and the ericaceous mycorrhizae also are very interesting. Ericaceous are the group that tend to hang out with plants that are in bogs and support their host in a very unique habitat. And then orchidaceous mycorrhizae are very interesting because the orchid seed will not germinate unless the right mycorrhizal fungi is present. Okay, so ecto and endo, I want to compare those two a little bit. And when you're studying this material, it'd be a good idea to kind of do a side-by-side comparison as you're studying to say, what's similar about these organisms and how they interact with the plant and the root, especially what's different about them. So think about those ideas.
Good question.
Yeah.
So for the orchidaceous
Mm-hmm.
mycorrhizae right here, so looking at this, are these both these last few ones that you mentioned, those are both in the boggy areas?
Orchids.
Yeah.
Not so much orchids.
Okay, where are those?
Orchids, well, unfortunately, we have to go to tropical areas to really see diverse orchids. Around here, we have lady slippers, and they tend to be on the forest floor, grow in forested systems on the, in the soil. But the vast majority of orchids actually are epiphytes. They like to hang out on trees in tropical systems. And so if you had the good fortune to go somewhere tropical and go walking out, look up, because you got to look up to see the orchids hanging out on the branches of the trees. And so they're called epiphytes. So if a plant grows on a plant, that plant that's growing on the plant is called an epiphyte, but they're not, the vast majority aren't harming the bigger plant, the tree. They're just hanging out on the tree to get better access to sun, but they're not causing any problems, except for there's a few exceptions that are pretty dramatic that are parasitic. But the vast majority of epiphytes are harmless to the host. So here we have two examples. Left is the VAM fungi. On the right is the ecto. And the artist did a pretty good job here showing some clear differences. So you see with the ecto mycorrhizal fungi, all this black on the surface of the root, that's fungal hyphae. And this word down here, mantle, I'd like you to know that word, please. So that's what they're describing. That thick growth of the fungus, of the fungal hyphae on the root is called a mantle. We call it the fungal mantle that's surrounding the root. And so this, if you think about it, this would provide good physical protection against something coming up to the root and trying to attack the root. Because the fungal hyphae, the way the artist drew it's pretty accurate. It's very, very compact. The hyphae strands, they grow really tight together. It's almost like a mushroom tissue look to it. When you dig up these roots and look at them, it looks like mushrooms you have on your pizza. Kind of looks like that much of a compact growth around the root. And you can see it with the naked eye. Once you train your eye, you can pick out ectomycorrhizal fungi growing on these roots. And so the fungal mantle, and then you can see that there are fungal hyphae growing off the root. Okay, that's really important that because otherwise, what's the point? The fungus, if it doesn't do that, then it's not really helping the plant too much. And so remember that the fungal hyphae, all mycorrhizal fungi, their hyphae extends well out into the soil. And not only can it extend out into the soil and, get water and phosphorus and nitrogen and potassium, but it can also link into other plants and trees nearby. And so it sets up this network of interacting plants and their fungal partners. And we're realizing how important that is to maintain a healthy forested ecosystem. The fungal hyphae are so, so important. And finally, that's getting into the macroecology world. The macroecologists are starting to realize this too. So it's a good time to be a microbial ecologist, because finally things are getting out there and getting to be more well known, and they're starting to apply this information. So fungal hyphae going off into the soil, which is great. And then you also see fungal hyphae going into the root tissue. So the black that you see in the root tissue, but you see how it's just surrounding the cells. And that's another word I'd like you to know, please. The Hartig net is right here, that word, Hartig net. So the Hartig net is named after Dr. Hartig. He's the one who visualized this first under the microscope. And just using an ordinary microscope like we have in our lab, you can see this. And what happens is the fungal hyphae just grows in the cell wall space. All right, so it's squeezed in between neighboring cells, right in the cell wall space, and that's where the fungal hyphae will invade the root tissue there. And that's important. It helps with nutrient exchange for both partners. Nice way to exchange nutrients. And then if you do see a fruiting body, a mushroom, around the base of a tree, you can bet it's most likely a ectomycorrhizal fungus that's doing that. And so that's just their way of going through sexual reproduction and then spreading via spores. Hopefully the spores get dispersed out into new habitats. Okay, now let's look on the left. Here we see the VAM, or the endomycorrhizal fungi. And the artist really didn't draw any thick mantle, which is correct. There is no thick mantle surrounding the root with a VAM. There is some, I wish they would have done a little bit more drawing on the root. There is some fungal hyphae that is onto the root surface, but it's very delicate, sparse, kind of, and so easy to disturb. Like if you dig up a root system that you're pretty sure has VAM fungi on it, you're going to rip all the fungal hyphae off the root. It's not thick enough to really persist on the root with all that disturbance. And so it's on there, but not nearly to the point of a mantle. But you still see the fungal hyphae growing off the root into the soil. And they also, the fungal hyphae of the endos can also hook into other host plants nearby. Then you see that the fungal hyphae can go into the roots tissue, and they're showing these weird little different looking growth patterns, and that's accurate. So the arbuscle term, I'd like you to know that. That's why they named it, that's part of the name. When you look that word up, it means dwarf tree. And that's what those things look like inside the root. So that's just fungal hyphae in the root, that once it gets into the root, it gets triggered to shift its morphology into these branches of fungal hyphae. So it does look, I'll show you a picture, a close-up, it does look like a little dwarf tree. And they just think it's a great surface area, right? Those short little branches like that of fungal hyphae really increases the surface area. So you got some great absorption going on there, an exchange of nutrients. And then the other term here that they put on there with, where is it? Vesicle here. I'd like you to know that as well. Vesicle, whoops, the vesicle term, and that is this balloon-like structure that you see. That's just fungal hyphae that is kind of morphed into this spherical balloon-like looking structure. And again, it's an increase in surface area, really helps with nutrient exchange between the partners. Now, the way the artist drew it, it's really hard to show it in any detail at this level of drawing, but you have to appreciate that the vesicles and arbuscles are not in the cytoplasm, just freely in the cytoplasm. What they do is they grow into the root tissue, the fungal hyphae penetrates into the root tissue, starts growing between the cell wall, and then it can get past the cell wall of the plant root cells, but then it butts into the membrane, the cell membrane of the plant, and that's as far as it goes, all right? And so it can't penetrate past the cell membrane because that would trigger a defense response by the plant. And so what happens is the fungus can push into the cell, like what the artist drew here, but the plant membrane kind of comes with it and keeps surrounding the fungal hyphae the whole time that it is in. So essentially, really, it's in between the cell wall and the cell membrane. Even though it looks like it's right in the middle of the cell, it's still being contained within the cell membrane. So again, really a little bit more intimate of a connection in that respect compared to the ecto, because the ecto just stay in the cell wall space. They don't move past that, whereas the endos can get a little bit more intimate and actually hug up to the cell membrane, so to speak. Here's some images of the ectomycorrhizal fungi. When you look at them under the light microscope, this is, so just using like a dissecting scope, they would look like this. And so most of the lighter colored stuff, like A here, that tan is the root, and then the black is the ectomycorrhizal fungi. That's actually a species called Cenococcum geophyllum. That's one that I worked with during my PhD work. And so one thing with ectomycorrhizal fungi, you can see this kind of Y-shaped pattern right here in E. It's really nice. Looks like the letter Y. So normally when plant roots grow, they just grow straight. They just, just a linear growth. But here you can see that the fungus actually, the interaction between the fungus and the root causes the root to actually shift its morphology into this Y shape. So if you ever dig up some roots that you suspect might have ectomycorrhizal fungi, look for that Y shape. That's a dead giveaway that you have ectomycorrhizal fungi. Not every single plant will do that, but the majority seem to, at least the ones I've witnessed with my own eyes. And so that Y shape is very common. And then you can start training your eye. Like I think maybe in panel A here, you can see how the root, that tannish root, is a little bit more slender. And then you see a kind of a thickening in diameter where the fungal mantle is. All right. So if the fungal mantle has grown up enough, you can see a shift in the diameter, and that even if it's the same color, I mean, here it's obvious that you got something else going on here because it's of the black color. But you can even see here in panel D, you can see that there is this thickening if you look carefully, and it does kind of look like mushroom-like material if you kind of take a needle and poke at it a little bit. So with ectomycorrhizal fungi, it's fairly easy once you train your eye to spot them. Even without a hand lens, it's always fun to take a hand lens when I'm out hiking. My boys used to get so frustrated with me because if there's a blown over tree, I'd start digging around in the roots and looking for mycorrhizal fungi. And they just weren't that into it back then. So, but
always, it can be like white too.
Yeah, yeah. It can be all different colors. I've seen pinkish, purpley, black, brown, white, gray. So it just kind of depends on the fungal species.
Because when I was doing like my little research project, we tipped over, there was like a dead tree.
Uh-huh.
It was like still standing, but we tipped it over, and all of the roots came out, and there's like this white spidery stuff all over, kind of like that.
Kind of like this?
Yeah.
Yep. So you were looking at ectomycorrhizal fungi, and yeah, especially if the fungus is a different color than the roots, it can really pop with the ectos. So this one in G, you can see really luxuriant growth, really nice colonization of the smaller roots. And so really fun to go hunting for that. Here they're showing, so here's a root, and then you can see the little Y-shaped small roots got colonized really well, and they morphed into that Y shape. And so this is Amanita muscaria. Does anyone know if you'd want to eat that? Not unless you want to go hallucinogenic. Can it make you go berserk? Doesn't Dr. Roberta say something about that in BI 112? That really? And then if you drank the urine, would you go hallucinogenic then? Yeah. So they were too chicken to like cook it up and eat it straight. They had to wait for the urine. Okay. I got it interesting. That reminds me, an interesting email just the other week. And it was, I mean, it ended, it was a happy ending, but it was really sad. A woman in the community, I think she was out in Gwyn, her dog, I think she said her dog was a lab or golden retriever, so bigger dog, not, you know, a tiny little thing. And it was out, you know, let the dog out in the yard to do its business and puked up some white mushrooms. And so she's describing this to me in the email. And then she said, and then the dog started stumbling around and acting like out of it, and then got really lethargic and kind of collapsed. And then one of their chickens, they had, you know, a bunch of chickens. Most of them were still roosting; they hadn't come out yet. But one had come out and was eating stuff in the yard, and that one died. And that was the only one that was out there, you know. So she's like, something's going on in the yard here. And so she grabbed her dog and took it to the vet. And then the vet was like, oh no, they were poo-pooing the idea of the mushrooms. And so they were running other tests. Well, as the dog was there, it puked up, you know, in mushrooms. And so white mushrooms. So right away I was, because we do have Amanita here in the area. And so I told her, you know, I forwarded her email to Dr. Robertus because he's the expert with looking at fruiting bodies. Since he's teaching that mycology class, he has years of experience with that. So I got to follow up with Dr. Robertus to see what he thought. But pretty sure that dog got hallucinogenic and really sick. Well, then they gave the dog activated charcoal at the vet to kind of sop up whatever was still in the dog, and the dog recovered. Thankfully, the poor chicken died, but the dog recovered. So again, you know, if you are interested in foraging for fungi, first of all, I would encourage you to take Dr. Robertus's class, and certainly don't go collecting mushrooms that you're not sure what it is. Amanita looks pretty, it's just a white, as far as I know, it's just pretty much white. It doesn't look like, if something's bright red, that kind of makes you think maybe I shouldn't eat it or touch it. But I think Amanita muscaria is all white, bright white. Right. And those are all Amanita species, different ones. So be really careful. Oh, and then the other thing too with foraging, even if you, let's say you get well trained and you're really confident and you go out and you're like, oh, I found some chanterelles or what's a better one? What's one that grows on the ground? Do they? Okay, so chanterelles. It might be a good idea to get to know that soil system a little bit, like the history of it. Like, did this used to be farmed? Was this a dumping site? Because heavy metals, if the main fear I have is that there's heavy metals in that area, in that soil. And then when you go, remember, they're good at sopping up heavy metals, and then they can retain the heavy metals in their tissues. So that makes me worry too. Don't know for sure. It sounds like a good strategy, but so the fruiting bodies actually can have high levels of the toxins. That would make sense. That would be a good way to get rid of it out of the hyphae that are doing so much work underground. You know, those hyphae have to be really healthy. Whereas the fruiting body, the only part that has to be healthy is where the spores are forming, really. So yeah, that would make sense. But yeah, so I I don't forage myself because I haven't had enough training to feel comfortable. But I do grow mushrooms in my house, and you know, you can buy kits and you can buy them. I have them sitting on the counter, and I'm still trying to figure out if anyone's taken Plant Kingdom lately. We always try to grow mushrooms in Plant Kingdom, and I usually pick lion's mane as one for the students to grow because it looks fantastic. It's a beautiful mushroom and it's super healthy. It's one of the healthiest mushrooms that they're aware of right now. Has all kinds of beneficial, even neurogenerative properties, they think. And so, and then what I'm struggling with is there's a recipe that I have, but I haven't done it correctly, obviously. But they say if you cook the lion's mane in using this recipe, it can taste like lobster meat. And I love the taste of lobster, but I don't like eating lobsters all the time. And so it's like, well, if I could get lion's mane to taste like lobster, that's like win, win, win all the way around. So if anyone can figure that one out, let me know. Because so far the recipes I've tried, it's like, nope, that's not lobster. Uh-uh. Shrimp? And they call it shrimp of the woods? Oh, weird. Oh, yeah. Oh, I'll have to look into that. I like shrimp too, but again, I don't eat much of that stuff anymore. So anyways, more pictures just showing some diversity. Here you can see there isn't huge Y-shaped morphology shifting going on. So you have to be careful. It's not always that way, but here that's just a fantastic picture looking at the real plump mantle surrounding the roots. And then I put this here again just to remind you, because a lot of these images, they're not showing the finer hyphae projecting off the mantle. So don't forget, there's always hyphae that come off. They washed all the finer hyphae away to get these images. But don't forget that that is going on. The hyphae go out into the soil. There's a lovely picture showing the fungal hyphae, how you have those very delicate, petite, small in diameter hyphal strands, much finer in diameter compared to the root system. So it's a real nice way for the plant not to have to invest in building finer roots, right? So the plant's not doing that. It's just partnering with the fungus and saying, okay, fungus, you do that. You grow your finer hyphae out and get access to that stuff. And so it's really conservation of resources in a way for the plant, because it doesn't have to invest in continually trying to build these small roots to get into those areas. It just lets the fungus do it. So this is Lacaria bicolor. And bicolor, two colors. So when you look at it in the woods, it's usually kind of a tannish. Sometimes to my eye, it looks a little faint, like pink hue to it, but tannish, I guess. And then if you look toward the bottom of the stem, it often looks purple. That image is a little grainy, but it's purple. And then when you grow it in the lab, this was one of my major fungal species that I worked with on my PhD. Beautiful purple color on a Petri plate. Really fun to work with in the lab. And then as the hyphae gets older, it'll start to turn tan on the plate. So you can see like the middle part is tan. The more vigorous growing part of the hyphae is still purple. So you get that bicolor look on a Petri plate as well. So Lacaria bicolor, I would like you to know that species name, please, because it is famous in the mycorrhizal research world. It was the first fungus to be completely sequenced, the DNA completely sequenced. Lots and lots of work. When I was working with Lacaria bicolor here at Michigan Tech University, one of my grad advisors was also working with a group out in France that were sequencing the Lacaria at the same time. So it was really kind of an exciting time to be working with that particular species. We chose it primarily not so much that the France group was already working on it. There were a lot of people working on it, but it's very common, especially here in the UP. You see Lacaria growing with a lot of the conifers around here. And we had, not me, but my grad advisor had gotten a big grant, and it was a consortium of scientists from different Midwest universities to look at, you probably never heard of the ELF antenna system that they installed when Sawyer Airport used to be KI Sawyer. It used to be a Air Force, sorry. The Air Force was there, very active base, and they decided to install ELF antenna systems, which are extremely low frequency antenna systems here in the UP. And these antenna systems were designed to communicate with submarines out in the North Atlantic. So I always thought that was a little bit of a head scratcher, but they claimed it would work. And so they had above ground antenna systems in the forested woods here. And then they had below ground. And then of course the question is, what impact does extremely low frequency electromagnetic radiation have on the environment? And of course, people were worried about human health. It was I mean, people were rioting. People were chaining themselves to the fences where these areas were fenced off. It was a big deal. It would hit the news all the time. I remember hearing about this. And so then we got a grant with a bunch of other universities to try to answer that question. What's going on if these get turned on? How does that impact the environment? And so that was what part of my research, that's how I got the funding to do my twist on that research. After years and years and years of studying it, I think it was a 5-year grant. The upshot was they could detect no discernible impact of those antenna systems on the environment. And so what they ended up doing is studying the fungi and the bacteria, because they're more sensitive to perturbations than trees. For a tree to display that it's being stressed, sometimes that can take years before you see any kind of stress symptoms, especially if it's just like a chronic low stress. So they opted not to really study the trees per se in the forested systems. They said, let's study the microbes that hang out with these trees. They're the ones that would probably show some huge shifts or die-offs or big shifts in populations or in ratios. And so we didn't see anything major happening. It was a lot of negative results, but from the perspective of the health of the system, that's what we wanted to see. But anyways, so it was a fun little thing to be involved in and to keep following what was going on. So this one, very popular around here. Lots of plants like to interact with it. And I picked it mainly also because it was relatively easy to work with in the lab. Some mycorrhizal fungi grow extremely slowly. This one was actually a fairly rapid grower. And so that was nice. It didn't take forever to do my experiments. So very, very interesting fungus. They know a lot, probably more about this fungus, actomycorrhizal fungus, than any other, because so many groups chose to work on it and get into it. It's a model fungus. So that's what it looks like when it's forming relationships. And then they've done all kinds of studies. This is from Nature, the journal Nature, where they're looking at of course they want to know, it's not going to let me do that, is it? They want to know how this whole partnership develops. So there's been, part of my research was looking at gene expression patterns as they change, before and after they're with the plant. And so it was lots and lots of work going on. How do they find each other? How do they communicate with each other? How do they set up this relationship? And because we want to learn more about that to see if we can help make that happen with other plants and make it happen more quickly and robustly to help plants grow in more marginal or not so nice areas. Okay, we got to stop there, I guess, and we'll shift to lab for the group 1.